Okay, so we just saw how in red blood cells, the hemoglobin that is normal hemoglobin is going to be globular, floating around, happy hydrophilic, all by itself, kind of in thousands of quantity inside the cytoplasm of the red blood cell, whereas the sickle cell hemoglobin is going to have these hydrophobic knobs that stick out, and then instead of being happy and hydrophilic, those are going to align together into a long structure that stretches out the red blood cell into that sickle shape. And this comes down to how amino acid R groups interact with each other and interact with the surrounding environment. The watery cytoplasm of the red blood cell was not conducive for those hydrophobic R groups in sickle cell to just float around by themselves. They were more comfortable slapped together than they were sort of putting oil into a watery environment. Whereas the hydrophilic R groups for the normal for the normal hemoglobin, we're quite happy to just interact with the watery cytoplasm. And so if we want to take away all of the details of the actual molecules of amino acids for a second and just think about their properties of being hydrophilic or hydrophobic, I like this example here for that reason. This little blue bit is the backbone. So you can imagine our polypeptide chain after translation. This blue bit here would be all of those amino groups, bound to central carbons, bound to carbox groups, all the way along. We could say, this is the N terminus and this is the C terminus at this end. And then color coding the R groups here in terms of whether they're hydrophilic or hydrophobic. So our pink R groups are going to be hydrophilic and our yellow R groups are going to be hydrophobic. And so if we took this baby protein, if we took this polypeptide chain, the product of translation, and then we drop it into a watery environment like the cytoplasm, it's going to fold based on how these R groups interact with the environment around itself. And so what you're going to see is all of those hydrophobic R groups kind of hiding with each other towards the inside, water-fearing, and all of these hydrophilic R groups happily pointing towards the outside because they're going to form hydrogen bonds with water. And so the three-dimensional shape of a protein, the overall shape that a protein ends up taking on, is determined by intermolecular interactions between the R groups of amino acids and also between those R groups and the surrounding environment, such as the cytoplasm. That's true in the cytosol, the watery environment of the cell, but that's also true in the membrane. If you think back to Unit 1 when we talked about the membrane is made up of phospholipids, but there's a whole bunch of stuff embedded in that membrane, and some of those things are proteins. And so when proteins are embedded in the membrane, they're going to take on a specific structure based on the hydrophobicity of the R groups, based on whether the R groups are made up of nonpolar covalent bonds and uncharged, or whether they're made up of polar covalent bonds or ionized and are partially or fully charged. So if we want to zoom in on this idea here, if we were to think of a lipid bilayer, remember that lipid bilayers are hydrophilic heads and hydrophobic tails. And so we've got hydrophilic, and then hydrophobic. And a protein is going to go all the way across the membrane. We would call a protein that goes through the membrane a transmembrane protein. Transmembrane means it traverses the membrane. So a protein that goes all the way across the membrane is still going to be made up of a chain of amino acids. And so this little stylized squiggly over here, I've represented on this side in individual circles that represent each amino acid instead of just a line here. And so you can see this whole chain of amino acids. Some of it is outside the cell, some of it is inside the cell, and then some of it is embedded in the actual membrane itself. Some of it is in the cytosol, this is outside the cell, so some of this is in the watery exterior of the cell. Some of it is in the cytosol, so the watery interior of the cell. And then some of it is embedded in these hydrophobic tails here. Some of it is embedded in this environment that is totally hydrophobic. And so what we end up seeing in proteins that are embedded in the membrane is that we have these amino acid chains where the majority of the amino acids that are inside or outside the cell are polar. The majority of the amino acids that are inside or outside the cell are made up of mostly nonpolar covalent bonds and are partially charged, whereas the majority of amino acids that are in the membrane itself, around those hydrophobic tails, are hydrophobic, are made up of nonpolar covalent bonds, or are neutrally charged. And so we kind of do like with like. Remember, like charges attract, and opposing like charges repel and opposing charges are attracted. And so if we go ahead and put a whole bunch of hydrophobic stuff together in the middle, they will happily hang out. And we can go ahead and put some partially charged stuff out here so it will form hydrogen bonds with water. This specific structure is important. These alpha helices that form down here in the middle, we'll talk about a little bit more in a later video.